Hi guys, hopefully we are fully functional. Okay, so in today's video we are going to go through the organelles, and primarily we're going to be relating structure to function for the organelles. Okay, I will periodically check the comments. Actually, right, just bear with me guys, let's get started here. So we'll be relating structure to function, so that's the main thing. So we will have a cell, and we'll have an animal cell. We'll be adding the organelles as we go along. And the main thing about the organelles is not just to know what their function is, not just to know what they're called, but to be able to relate their structure to their functions. So that's very important, because the structure is essentially the way the structure is the way the cell is adapted to, or the structure is the way that organelle is adapted to perform its function. So to be able to relate its structure to how it performs its function is very important. Okay, so guys, let me know if the video is okay and if the sound is okay, and if that is so, I will continue. So let's proceed. Okay, so let's carry on. So we're going to start with the nucleus. So the nucleus is here, or wherever. And what are its key structural features that are, or what is function? Let's begin with its function. So the function of the nucleus is essentially to control the activities of the cell. You knew that at GCSE, but what is important at this level is to understand how it controls all the activities of the cell, and how its structure is related to it being able to do that. Okay, so key structural or key things about the nucleus is that it contains the genomic information of the cell in the form of DNA, that the DNA in an animal cell or a eukaryotic cell is going to be associated with histone proteins, and at times of division the DNA will condense into visible, or at least visible under the microscope, chromosomes. Okay, so those chromosomes are always there, these long molecules of DNA, always there, however condensed at the time of division. Okay, so remember, any questions, do ask. Okay, so let us proceed then. Okay, now the function, so you've got the DNA. The DNA is organized into chromosomes, and the chromosomes contain the genes, and the genes encode for proteins, and it's the proteins that are going to be the machines that will allow the cell to carry out its functions. Okay, so next, within the nucleus, so there's a few structures here that we need to be aware of. So first, we've got the nuclear pores of the nucleus. Now they allow mRNA to come out of the cell because remember the DNA molecules are too large. There's too much information stored in the DNA for all of it to come out just to synthesize a protein. So you've got the nuclear pores. The nuclear pores allow messenger RNA molecules to exit the nucleus. Okay, so you've got the DNA in here. Now in the DNA, or in the nucleus, you have these regions which are very active in terms of gene expression, and these regions are called the nucleolus. Okay, so within the DNA, you have these distinguishable bits, a bit lighter or a bit darker, however you're seeing it. But in these areas, there's lots of active gene expression going on, and this is where the genes for the ribosomes are being transcribed to produce the ribosomal RNA and the ribosomal proteins needed to synthesize the ribosomes. Okay, so that is the nucleolus area. So within the nucleus, we have the nucleoli, or the nucleolus, and these regions are where the ribosomal RNAs are being transcribed. Okay, so next, let's say something is transcribed and it moves out of the nuclear pore. The next place that it will go to is the rough endoplasmic reticulum. Okay, so these nuclear pores are structures, and it's important to note that actually the nucleus is a double membrane structure. So let's just redraw that. So one membrane and two membranes, and this is now the nuclear envelope. The nuclear envelope has pores. Okay, so let's just draw a few nuclear pores, and these are the things through which the mRNA might move out of the nucleus. Okay, so we have these pores, like so. So there's two membranes that go around. So we say that the nucleus is an enveloped organelle. It's an enveloped organelle. Envelope refers to an organelle that has two membranes covering it or containing it. Okay, so we'll think of the nucleus like this. You've got the DNA inside, so the majority of the genomic information in that chromatin form associated with histone proteins. The chromosomes are there, but they're not always condensed, so you can't see them in their tradition or the highly popularized X shape. You don't see that. That only happens during division. Okay, but then you've got these regions of highly active gene transcription going on called the nucleolus, and they're highly active because it's the ribosomes that are being synthesized there, or at least the ribosomal RNA, the ribosomal proteins. Okay, so the next structure that we will encounter will be the rough endoplasmic reticulum. So what we'll do is we'll just follow along the journey of the protein synthesis. And so the next thing that will happen is that this membrane itself extends out to form the endoplasmic reticulum. Like so. I won't draw it too complicated. But something like that. Okay, so it's the membrane of the nucleus that actually extends out to form the rough endoplasmic reticulum. And the structure of the rough endoplasmic reticulum is to be described as a series of interconnected, flattened, membrane-bound sacs. And they have ribosomes attached on their surface. I'm not going to draw all of them, okay? But the ribosomes is what allows these structures, or the rough endoplasmic reticulum, to be involved in protein synthesis. So an mRNA will leave the nucleus through the nuclear pore, okay? And as it comes out of the nuclear pore, it can attach to one of these ribosomes, and the protein synthesis can begin into the cisternae of the rough endoplasmic reticulum. Okay. Now, one of the key roles of the rough endoplasmic reticulum is to enable the polypeptide to become folded into its tertiary structure. So, in the rough endoplasmic reticulum, there are proteins, there are enzymes involved in helping the polypeptide synthesized at the ribosome to adopt its tertiary or secondary and tertiary structure, as well as some enzymes that could be involved in modifying the protein slightly. Okay, now the next structure that we'll look at is the next part of the story of protein synthesis. So when the protein has adopted its tertiary structure, it then moves, so you might get a polypeptide synthesized here. As it moves through the rough endoplasmic reticulum, it will start to fold up into its tertiary structure, and it will bud off of the endoplasmic reticulum in a vesicle. Whoops. In a vesicle. Okay? And we'll end up in a situation where that vesicle, man, I'm getting the hang of this, where that vesicle is going to be moving towards the Golgi apparatus with that protein contained. Then it will fuse. It will fuse with the Golgi apparatus. So the Golgi apparatus is a series of flattened sacs which are not interconnected. Okay, so that's the description, right? A series of flattened sacs, also called cisternae, but they are not interconnected. So careful of that when you are describing this organelle. Okay, but remember the function of the Golgi apparatus is to modify proteins synthesized in the rough endoplasmic reticulum, modify those proteins and package them into vesicles, and those vesicles then move to other parts of the cell. It could be the cell membrane, it could be other organelles that require the protein synthesized, and so on. Okay, so what will happen is that these vesicles, so this vesicle will arrive at this face. I don't remember if it's the cis face, I think it's the cis face of the Golgi apparatus with that protein contained, and that vesicle will fuse with the cis face of the Golgi apparatus, and once added in to those membrane sacs, then enzymes within the Golgi apparatus then continue the modification process. So they will add carbohydrate to these proteins modifying them, and these proteins then move through. So it buds off of one, fuses with the other, buds off from one, fuses with the other, until it gets to the end, where its modification should at this point be complete. Okay, so it gets there, and then at this point its modification is complete. It buds off of here in a vesicle. Now this vesicle can then go to where it needs to in the cell. So there are maybe a couple of locations that you need to be aware of. Okay, primarily proteins synthesized in this way are going to be secreted from the cell. So this vesicle will go towards the cell membrane. It will then fuse with the cell membrane, and the contents will be ejected from the cell via exocytosis. But it could also be that this vesicle is going to another organelle, another organelle called a lysosome, right? So the lysosome has got lots of hydrolytic enzymes. Okay, so it's got some lipases, for example. It might have some proteases. It might have some carbohydrases. So all these enzymes. And it might be that the protein that was synthesized via this process, it might be that the protein that was synthesized was a hydrolytic enzyme destined to be added to the hydrolytic enzyme content of this lysosome. And so it might be that the destination of this vesicle is actually to go ahead and fuse with this lysosome, thereby delivering this enzyme to that lysosome. So that could equally have been its destination also. Okay, so just to be aware of that. So while we're here then, we might as well talk about the lysosomes, which are membrane-bound organelles. In terms of their structure, they contain hydrolytic enzymes, and so any component in the cell that needs to be degraded or broken down to its constituent monomers for the monomers to be reused again, those proteins or those organelles might be sent to the lysosome, fuse with the lysosome so that the enzymes inside can degrade them, hydrolyze them back down to their monomers. Okay, so let's move on. Okay, what have I missed? Right then, let's continue. So that's the protein synthesis machinery. But while we're here, let's just take care of this other kind of reticulum called the smooth endoplasmic reticulum. Now, the smooth endoplasmic reticulum doesn't parallel completely the structure of the rough endoplasmic reticulum. It has a more kind of bulbous appearance, but essentially it's got a very similar structure to, or in nature it's similar to the endoplasmic reticulum. So these are interconnected in a way that slightly have a different appearance under the microscope. Okay, but the smooth endoplasmic reticulum, so let's just label these, rough endoplasmic reticulum, the Golgi apparatus, we have lysosomes, okay, and we have the smooth endoplasmic reticulum. Now the smooth endoplasmic reticulum is, I guess we could still call them a series of interconnected membrane sacs, but maybe we don't have to refer to them as flattened membrane sacs because they're not so flattened. In terms of their structure, they are very similar. They are membranous sacs, but they contain likely different enzymes because remember the function of the smooth endoplasmic reticulum is for the synthesis of steroids and lipids. So all the lipid synthesis is going to be happening here. So this organelle will need different enzymes in order to carry that out. Okay, and it's worth noting that any enzyme that's synthesized for working in the smooth endoplasmic reticulum would have come from the Golgi apparatus. So if the smooth endoplasmic reticulum needs an enzyme, then that enzyme ends up at the smooth endoplasmic reticulum because it was synthesized by the Golgi apparatus, and that enzyme is delivered there via a vesicle. Okay, so that is that. Okay, so we have the smooth endoplasmic reticulum. Now, while all this is going on, and I guess we should have talked about it while we were discussing the protein synthesis machinery, that the thing that allows these vesicles to be transported from one place to another is the cytoskeleton. So the cytoskeleton is these proteins that form filaments through the cell, okay, forming a network of fibers upon which the vesicles can be transported from one place to another. So the vesicles are not just floating around, zooming from place to place according to any will of their own, but what is happening is that there are mechanisms that allow vesicles to be moved along these cytoskeletal filaments within the cell. And it's along these filaments that vesicles can be transported. These filaments also allow the cell to have some form of shape that is regulated. And these filaments are also involved in perhaps moving some cells from one place to another if that's part of their function. Okay, so we have the cytoskeleton right there, filament, likely made of, usually they're made of proteins which are insoluble. So these, or actually no, they are soluble proteins, but they can associate with each other to form these filaments. And by regulating if they are in their soluble phase or if they're in their filament phase, the cell can control the synthesis of these structures. Okay, right, so we've had lysosomes. Okay, right, while we're on the cytoskeleton, I should not forget that one of the important cytoskeletal components is microtubules. Now, microtubules have all the functions that I have talked about so far, but remember that at their center, they have the microtubule organizing center, or centrioles. Okay, and remember that the centrioles are the structures that allow the formation of the spindle fibers during mitosis. So do not forget those. Okay, moving on then, we have flagella and cilia. Now, in an animal cell such as this, it's quite rare to see these structures, but they are possible. Okay, so what we might do is just move that off for a bit. Don't mislead anyone. Okay. So let's look at a flagella. Now, flagella and cilia are both structures based on microtubules. So, but they have slightly different functions and slightly different structures. Okay, so generally speaking, both cilia and flagella are made of microtubules, essentially. So it's these microtubule fibers that form these structures that poke out of the cell. So if we have a cell here, a cell there, we have a cilia, okay, and we might have a flagella, okay. Now, cilia and flagella both based on microtubules, but they have slightly different structure and slightly different function. Okay, now while cilia are involved mainly in moving liquid across the cell, such as you might find in the epithelium of the lungs. So while cilia have that kind of function, they also have a sensory function, right? So they have sensory and they move fluid. They're not moving the cell, they are moving the fluid around the cell, and they have a sensory function. Okay, so as the fluid might move over a cell, it might cause a cilia to move, and in such case, then the signals get transmitted inside the cell, allowing the cell to respond to that. So cilia move fluid in some cells, and in some other cells they have a sensory function, whereas flagella usually are longer in terms of their structure. In terms of their function is usually to move. So in most cases, the function is to propel the cell forward or move the cell. However, they can have some sensory functions as well, but that's the main difference between cilia and flagella. Okay, coming back to our cell then, which organelles have I forgotten? So we have mitochondria next. Okay, so we might have a mitochondria like so. Again, this is an enveloped organelle because it's got an outer membrane. It's got a highly folded inner membrane, which fold to form cristae. Okay, and it has these stalked particles visible on the inner membrane, which are not ribosomes, but they are, as year 2 students will know, they are the ATP synthase complex, which is in that inner mitochondrial membrane. Okay, so we have the inner mitochondrial membrane, we have the outer mitochondrial membrane, and on the inside here we have the matrix, and we have these structures called cristae, right? And we have the stalked particles right here, stalked particles, which are the ATP synthase complex. Now, in terms of the structure, remember, well, those are all structural features. Remember that there is this concept of surface area, and if the inner membrane of the mitochondria has stalked particles, has the electron carriers to carry out the oxidative phosphorylation stage of respiration, the more surface area we have to contain those components, the more efficiently the mitochondria can work, and it can carry out respiration at higher rates. And remember that in order for the matrix to carry out the Krebs cycle stage, the link reaction stage of respiration, it has to have all the enzymes in order to do that. So remember that that is happening there. What else? Ribosomes, Golgi apparatus, protein synthesis. Okay, right. Right, so what we'll do now is we will move on to chloroplasts next. So we're going to move away from this animal cell, though remember that plant cells have all these organelles, but in addition, it has others also. Okay, so we will now move on and look at plant cell. So the plant cell has on the outside, it has that cell wall that is made of cellulose, and the cellulose cell wall is obviously it's composed of cellulose. That is a structural detail, So we have cell wall right there. Key points about that is that it is made of cellulose. Functionally speaking, it is important that cellulose is both able to support the cell as well as be permeable to solutes and water, which it is able to do because of its structure. Okay, so that's important there. Then we have the inside, we have the vacuole, okay, and that membrane, which is called the tonoplast. Okay, and we also have a nucleus, which roughly, well, it does perform the same function as we have, but let's concentrate on the things that are unique to the plant cell. So we have the cellulose cell wall. Okay, we have the vacuole with the tonoplast. That is important in storing fluid for the cell. And we are going to have chloroplasts. Okay, so chloroplasts, zoom into the chloroplasts. Now chloroplasts have, again, they're an enveloped organelle. They have an outer membrane, and they have an inner chloroplast membrane. But then they have this fluid structure inside called the stroma that contains the enzymes for the light-independent stage of photosynthesis, as well as containing the DNA of the chloroplast. But on the inside, it's got these membranous structures called thylakoids. Okay. And in many ways, these are parallel to the inner mitochondrial membrane because, again, they have electron carriers, and it's here that the light dependent stage of photosynthesis happens. So these thylakoids contain the electron carriers and proteins needed to carry out the light dependent stage here. The stroma has got the enzymes to carry out the light independent stage of photosynthesis. Again, we have this structure in order to maximize the surface area of the thylakoid membranes, so we can contain more of these electron carriers and proteins to increase the efficiency with which light can be captured by the photosynthetic pigments, and therefore the efficiency of the or the productivity of the plant can be maximized. Okay. Right. So what else? Okay, I think that I will call it a day there. And we will continue next time with, what shall we do next time? We'll have a bit of comparison of the prokaryotes and eukaryotes, and maybe we might even make a start on the biological molecules. Okay guys, thank you very much for tuning in. I will see you tomorrow. Tomorrow I will likely be doing the stream at 3 o'clock rather than 4 o'clock, so just so you know.